Hello, everyone. My name is Tiffany Drummond, and welcome to today's program. We're talking about some very exciting developments in MPN treatment, including important breakthroughs and promising new combination therapies. Many of these advancements were recently highlighted at the American Society of Hematology meeting, better known as ASH, where leading doctors and researchers from around the world gather to share the latest findings. As a clinical research and patient advocate, I am excited to tune in. The patient stories goal today and every day is to provide you, patients, caregivers, and partners with valuable information to help you in your healthcare journey. We want to empower you to have informed conversations with your medical team so you can better understand your treatment options and how to balance effective care with maintaining your quality of life. Before we get to our speakers, we want to thank our sponsors, Sobi and Insight, for their support, which helps us to host more of these programs for free to our audience. The patient story retains full editorial control over all content, as always. I must also thank all of our promotional partners for their support. It is because of you our programming reaches the audience who needs it. I hope you find today's program helpful, but please keep in mind that information provided is not a substitute for medical advice. Finally, we know there's a lot of information that you want to know more about and discuss or at least tune into, so please let us know what those are, along with what we can do better to make sure we provide that for you. A survey will pop up after the program ends, so make sure you don't go anywhere. Let's kick off another engaging conversation moderated by an amazing patient advocate, Andrew Schorr, and leading hematologist oncologist, Dr. John Mascarenhas. Take it away, Andrew.
Hello, and welcome to this program about the latest in MPNs and more specifically myelofibrosis. I'm Andrew Schorr. I'm with a friend and a leading scientist physician. I'm in Los Angeles. He's in New York. There's Dr. John Mascarenhas at the Tisch Cancer Institute at Mount Sinai in New York. And John, you have many titles. I know you're a noted hematologist and a subspecialist in MPNs. Thanks for joining us.
Yeah, Andy, thanks for inviting me. I always enjoy connecting with you.
Well, as you know, it's very personal for me, and I'll mention to the audience, I've been living with primary myelofibrosis since 2011, and it's progressed some. So I've been on first one JAK inhibitor and then another JAK inhibitor. Maybe I'll switch to a third JAK inhibitor. Will I have combination therapy with a JAK inhibitor and something else? We all wonder about that. Some of us are concerned, should we have a transplant, or can medical therapies take the place of transplant? If you have polycythemia vera, or if you have essential thrombocythemia, you say, well, am I going to progress to myelofibrosis, and at what rate? And for any of us is, how is our situation different from the next person? Now, John, as you know, you were a speaker at really, I call it sort of the World Series of blood conditions and blood cancers, the American Society of Hematology meeting that was in December in San Diego. And you were involved in lots of commentary or studies that you were involved in. And so what I want to talk about with you over the next hour or so is what's significant for patients. Because we have the current therapies and we have, I don't want to say an alphabet soup, but we have a lot of stuff, drugs that many of us have never heard of that are in development, and you're involved in a lot of the development. Which way is the wind blowing? So let me start with that, the 10,000 foot level first. You go to this conference and day to day, you're both with patients and you're looking at the science. Are you encouraged for us? Because we saw progress in other blood cancers. Is it now starting to blossom in MPNs and specifically for myelofibrosis?
Yeah, I mean, so I think in short, the answer is yes, I am encouraged. I think that's a fundamental defect that I have, that I continue to be optimistic no matter what we're looking at. And that optimism, has been maintained over almost 20 years at this point, that we continue to move in the right direction. Unfortunately, often not fast enough for our patients. But what I've seen is the evolution of, the JAK inhibitor era, which you alluded to, where we have 4 JAK inhibitors that are approved that allow us to tailor and personalize the therapy to patients based on their patient profile and their blood counts, and even provide serial sequencing of JAK inhibitors. But that isn't enough. So there's been a lot of interest as we understand the disease biology even greater than we did in 2005 when JAK2 mutation was first discovered. We now recognize that there's a greater degree of complexity and heterogeneity amongst patients. So there are a lot of different pathways that do appear to be very important and relevant to the physiology and the pathophysiology of this disease. And there is a real interest in targeting the malignant stem cell, that grandmother cell that originated the disease, in which we are looking for those vulnerabilities in those pathways in that cell population that would allow us to ultimately delete that cell to provide deeper responses, even at what I would say would be curative approaches. Because outside of transplantation, the therapies we have don't actually cure patients. They do address issues, which are not unimportant, whether it's symptoms, spleen, cytopenias, low blood counts. And I do think that doing that improves how patients do and ultimately prolongs survival. But what we're really looking to do is use these findings, leverage these findings from the lab to find therapies that actually change the disease course, that can improve outcomes like survival and progression-free survival. So there are many agents that I think are leading us in that direction. These are agents that turn on the p53 pathway. So drugs like navtemadlin. These are oral drugs that bind a protein called MDM2 and relieve repression. So it's a double negative. They relieve repression on p53, allowing for the natural cell processes to be induced to commit suicide, cell death of the malignant cell. So it's really a fascinating concept. Navtemadlin is at the forefront of doing that. We showed data in the relapse refractory setting of using that agent as a single agent. We're now going to move it up to combinations. And drugs like that, and I'm using that as an example, I think are telling. It's hitting pathways that can induce these malignant cells at their core to die, to synergize and be practical with it. We want to create therapies and approaches that capitalize off the benefits we have, like JAK inhibitors, that can be well tolerated, but can provide deeper responses than what we've seen thus far. So navtemadlin, I think, is a great example of that.
All right. Well, we're going to go through sort of a little bit of a laundry list as we dig into it of different drugs that have been mentioned. Some of us have never heard of them, but I just want to go over what you said so I understand. So first, you're trying to go back to the very basics of cancer. What went haywire in a patient who ends up with a bone marrow problem, right? That leads to one of the MPNs, and can you shut that down at the earliest stage by understanding it? Now, over the last few years, your scientific community has identified different oncogenes, cancer genes, that have been responsible for that. And you talked about the heterogeneity, the differences of us. Some of us have CALR, some of us have JAK2, some of us have MIPL, etc. Is that understanding helping?
Well, I do think it helps because we recognize that it's not a monolithic disease, and that the different mutations that we call the driver mutations and the different amounts of those mutations that are understood to be present in the bone marrow cells, as well as the sequence in which the mutations arose, all tell a picture. It paints a picture of complexity at the molecular and cellular level that explains why there's heterogeneity at the clinical level, why some patients have very high white counts and some patients have very low platelet counts. Some patients have very big spleens, and some patients have a lot more anemia and transfusion dependence. So it can all be explained relatively to the biology, to these mutations and to the effects of these mutations. The next step, once we've realized that, which I think we've come to that realization, is how do you take all that complex data, and it's a lot of complex data, and that's where things like maybe artificial intelligence and machine learning will help us move the field forward, as it's doing with other sciences. And how do we distill that to help us understand how best do we then target those cells based on that complexity, that genomic complexity. So I do think we're moving in this direction of deeper understanding of the biology from the molecular standpoint. That informs us with both prognostication, which is important, but most importantly, I think therapeutic implications. So I'll give you an example of where I think that might make that might make sense. There is substantial. So, for example, there is substantial data that would suggest that certain mutations, likely influence outcomes and responses to treatment. For example, if you have high molecular risk mutations, these could be mutations in ASXL1, EZH2, IDH, mutations that some patients may have heard of and even have seen on their reports and can ask their physicians, do I have these? Because most patients will have had NGS, next generation sequencing. So looking at these gene panels and seeing, do mutations exist in any given individual? And what does that mean? Well, we know that some of these mutations can have influence prognosis and outcome. And some of these mutations and the presence of them, even more than one mutation, could even predict for a lesser response to drugs like ruxolitinib, a lesser robust spleen and symptom response, a shorter duration of response, a quicker time to failure of drugs like that. So knowing that up front may be strategic in understanding how better to approach diseases rather than give the same drug for everybody. Like maybe the same drug for everyone is not going to be the right answer. And I think at the forefront, drugs that target mutant CALR, for example, I think are really exciting. This idea that we're taking out a subset of patients with myelofibrosis and ET that have the mutant CALR protein that's expressed on the surface of the cell, and we're saying, well, these patients may be best served by a drug that specifically targets that protein on the surface. That wouldn't make sense for a JAK2 mutated patient. A JAK2 mutated patient may be best served with a small molecule inhibitor that specifically and selectively targets the JAK2 mutation, which is under investigation. So you're seeing these mutations inform the clinical development of very selective and specific drugs.
Okay, so I want to do a little promo for patients now, and that is wherever you may be watching this, I think this advocacy for next generation sequencing, NGS as you referred to, so that a picture of, I almost think of it like a modern art painting with red and blue and splattered over here and stuff like that. What are your colors, right? And then hopefully with some consultation with an MPN specialist, how does either the current therapies or maybe in a clinical trial, investigational ones, apply to your specific situation at that time? And I think you also mentioned, John, how the genes that are driving our illness may evolve. So what the story is today may not be the story in a year or two or three. I've been living with myelofibrosis since 2011. It has been pretty stable, and it has been driven by the JAK2 V617F. Now, that brings us to current therapies, John. You mentioned at the beginning about the 4 current approved JAK inhibitors. They have nuances, and it would seem like the choice of which one or sequencing, as you said, may vary by patient based on their situation, right? But I mean, we have good drugs. It's like, where do you start? And then we'll go on with our discussion of building on that as far as combinations. So how do you know where to start?
We are blessed that we have choices today because it wasn't always the case. So I think what's nice is that we can have opportunities to select drugs that may be best suited for different subsets of patients. So for example, patients with low platelets, what we call the cytopenic profile of patients. These could be patients with platelets of 50,000 or even less, or even patients between 50 and 100,000. I would say low platelets less than 100,000 may be best served by drugs that are easily delivered and have rationale in that patient population, namely pacritinib over ruxolitinib, in which we know platelets often limit the ability to dose up on ruxolitinib. Fedratinib even more recently had some data in the patient platelets between 50 to 100,000, providing some more security there. So we know that platelets could be a determinant of which one of the JAK inhibitors you're going to select. Anemia is another one that's gotten a lot of attention because anemia can be a major issue for patients at presentation during their clinical course and can worsen over time. And that ruxolitinib, great drug, can worsen anemia for some patients. It may not be the best therapy in that setting. So drugs like momelotinib or even pacritinib that inhibit ACVR1, a different protein, can improve anemia in some patients. So we use the patient profiles to help us understand maybe which drugs to choose from. But I will say that for the most part, most patients will start with ruxolitinib. It is the oldest, it's the most familiar, it is probably still one of the best drugs that I've ever seen in this field in terms of achieving its goals. But again, not every patient fits the bill, and you can sort of tweak that as needed.
At ASH, and I think a lot of studies during the year talk about building on ruxolitinib, Jakafi or Jakavi in Europe. And I'm sure there's discussion about building on one of these other JAK inhibitors as well. So is a 1-2 punch necessarily better? Is that where you're headed?
Well, it's definitely what we're interested in asking. So the question that is being asked in a lot of the clinical trials, particularly these mature clinical trials, is simply we have these JAK inhibitors that are disposable and they're beneficial, but they're not sufficient. We have other drugs that have now demonstrated clinical activity and are rational and are validated in preclinical models, so systems that will help us understand whether it makes sense to take it into a patient. And these are drugs like pelabresib and imetelstat and nabtemadilin and selinexor, all funny sounding names, but each drug has rationale and each drug is active, even as a single agent in these diseases. But the questions that are being asked now, is it better to combine the two drugs? If you look throughout oncology, most of oncology is treated with combinations of therapy. It's very rare to find an oncologic disease, a malignant disease, whether it's of the blood or of the solid malignancy, where we use one agent, and then if it fails, we go to a single subsequent agent. Usually it's combinations of therapy that are more potent together. A term we often use is synergize. Suppose I know that everyone understands what that word really means. It really means that if you take either drug alone, you can do this in a lab, and you expose them to malignant cells, that the combination of the two drugs works better than 1+1. It's almost 1+1+1. You get even better effects in terms of killing or limiting those cells, those malignant cells. So we hope to replicate what we see in the lab or in mice that are engineered to have these diseases in humans. And the data has taken us, the field has taken us towards the route of combining these novel agents that have shown activity in the relapse refractory setting with as single agents as combinations up front. And most of the data would point to that if you put a agent X, whether it's selinexor or nabtemadilin or pelabresib or imetelstat together with ruxolitinib tends to be the first drug to pick, that you tend to see better efficacy, in some cases even better safety profile when the two are combined. So the natural question is, can we take two active agents? Can we get deeper responses? And you may ask yourself, well, what does deeper response mean? Well, it could mean at the very most superficial, more spleen reduction, that's a regulatory endpoint, deeper symptom improvements. But we're looking at other biomarkers to understand, are we really hitting the target that we want? Are we getting deeper reductions in the driver mutation amounts of what we call the VAF, the variant allele frequency? We can measure these. Are we reducing those numbers to suggest that we're reducing the burden of disease in the bone marrow, since we can't measure it with a CAT scan? Are we reducing the disease in the bone marrow from other viewpoints, like fibrosis? Is the amount of fibrosis reducing? Are the amount of circulating abnormal cells, are they reducing, something we call CD34 cell number? Are we also reducing cytokines, inflammatory markers to a deeper extent? So we look at all of these biological aspects and hallmarks of the disease, and we ask, are we getting even more profound effects on these biomarkers, suggesting that we're really modulating the disease more effectively? At the end of the day, MPN patients want to live better and longer. But we look for markers early in trials to understand, are we getting there? Rather than wasting people's time, are we going to achieve?
Okay, so if 1+1 can equal 3, what we say as patients, we say, well, doctor, these are very powerful medicines. Will my quality of life be diminished if you add this other big gun? Because after all, we want to live well. We want to live longer for sure, but we want to live well. So could you talk about the power of the combinations, but the worry about additional side effects?
Absolutely. I'll give you a prime example where 1 plus 1 can equal 3 from an efficacy standpoint, but might still equal 1 from a safety standpoint. So a great example of that is the Manifest-2 study, where we took JAK inhibitor naive patients and with myelofibrosis, and we randomized them to standard of care that would be ruxolitinib plus a placebo. That's double blinded, so the patients and the investigators don't know what that person is getting, only a computer knows, versus ruxolitinib, which is Jakafi, plus the study drug pelabresib, which is an oral BET inhibitor, a very rational drug, which modeling has shown us should synergize very nicely with Jakafi. And the answer is it did, efficacy-wise. If you look at 24 weeks, very profound reductions in spleen, and I would argue very profound reductions in symptoms. And if you look at the biomarkers, the bone marrow fibrosis reduction was far more significant. The reductions in inflammatory cytokines, the reductions in JAK2 mutation, everything aligned with superiority of the two. But most intriguingly, if you looked at the safety, there was less grade 3, 4 treatment emergent side effects with the combination than with the single agent. And I've never seen that before, really, where you took two active agents, you combined them, you got almost double the clinical activity, and you got less toxicity. So there's a great example, and what I hope is reassuring for patients that double the action doesn't mean double the trouble. You can actually combine some of these drugs and get good activity and not make patients feel worse, but maybe even make patients feel better and have less toxicity. So that's one example. The other thing that I think we have to acknowledge is that we do add toxicity sometimes when we add combinations, and sometimes that toxicity is in the form of gastrointestinal GI toxicity, and sometimes that toxicity is in the form of lower blood counts, and sometimes it's a trade-off. Are we getting deeper responses that could lead to better outcomes where we could be getting more cytopenias, more lower counts, and more need for monitoring and even transfusions? Is that reasonable for a given individual? Or we could be adding some nausea and diarrhea by doing that. Key to this conversation is, are we adding these toxicities continuously or periodically when some of these drugs are dosed? So for example, navtemadlin, very active drug. When we looked at the data, it was very clear you could get, and about half the patients did, some GI toxicity. It was mostly low grade and easy to manage, but it's there. But the drug is dosed for 7 days in a row out of a 28 day. So it's 1 week and 3 weeks off. And the toxicity is mostly relegated to day 2 and 9. So the question, and this is a esoteric or personal question, is for any given individual, if that is a deeper response that could lead to better outcomes for the patient, is that period where you might have a week of not feeling optimal and maybe having GI toxicity, is that worth it? And I don't know. I mean, that's from a human perspective. I'm not really sure. I mean, I know from a clinical investigation, we're interested in trying to understand, are we providing full good at a price, or is it going to be good and no price? I mean, nothing comes for free, but these are questions that are important, and I think we rely on the patient community to tell us actually. So I mean, we do patient global impression of change, which I think is a really important tool. I want to stress that for a second. So we don't just ask patients, how are you feeling, and doing the myelofibrosis symptom assessment form, but there's also a tool that we use, very validated tool, called patient global impression of change, which is really simple. It's asking a question, putting everything together, all the toxicities that might ensue, maybe benefits that you're noticing or just being told that you're getting from the physician. If you put the whole thing together, do you feel like you are the same, a little better, a lot better, a hell of a lot better, or in the reverse, a little worse? And that's a very valuable tool, because patients will tell you if the whole thing is worth it or not. So that's really key, I think, is to making sure that those align, that the patients reporting is that they're globally, that they believe, that they feel that what they're doing, what they're going through at the end of the day is a net benefit.
All right. I want to go down the road of discussion about transplant, where we are in 2025 for a second. So, and we're going to talk more, and you've mentioned a number of these drugs that are investigational right now on top of the 4 approved JAK inhibitors. So there are patients younger than me. I'm 74. so I don't plan to do it. But there are patients younger where it's been recommended they have a transplant. And as you say, it can be curative. It is a big gun. And I lived for a long time in Seattle where they developed it originally. And I knew about the morbidity and mortality, and that continues, I think, for some people. So where are we now with transplant versus all these pills or medicines administered in a less big way, if you will?
So transplant remains the only modality that we have, fortunately or unfortunately, for cure. And as you've pointed out, it's not for everybody. So if you're advanced in age or you have too many comorbidities, other illnesses, transplant may be more dangerous and detrimental than it ever will be helpful. So it's for a select group of patients, and even those patients that go into transplant, which I am an advocate for, are moving into an aggressive type of therapy, but it has to meet the aggressive nature of the disease. So you would never take someone who has a very good version or low risk version of the disease right into transplant, because you could cause more harm earlier on than good. So it really is a complicated, discussion that involves understanding where the patient is from a disease perspective, the nature of their disease, their goals and understandings and expectations, and making sure they have a donor and support system to do a transplant. So I do encourage that conversation. I think it's important, but it's not going to be for everyone. My hope, my goal would be in my lifetime, in my career, is that we ultimately develop therapies that make transplant unnecessary, that we can develop medicines that, because what transplant really is at its core is taking immune cells, someone else's immune cells, in fact, and using them to ultimately get rid of the grandfather, grandmother cell, what we call the stem cell that started the disease process. And that's an immune mediated elimination of the cell. So if we can figure out how best to do that with medicinal therapies that may not be as intense, then I do think we could get to a point where transplant may become historic, and maybe we really don't need transplant if we can go after those originator cells. And that's really what a lot of here at Mount Sinai, for example, that's what a lot of our translational research has been based on. And this is something that has been taught to me by Ron Hoffman and others. It's really a stem cell directed approach to shut down or eliminate that pool of cells that allows the disease to persist even after you can wipe out cells with a transplant, that those cells can come back. So it's really targeting the stem cells with therapies that are rational. And the only way we're going to do that is by using science and collaborating with patients. Because I have to tell you, if it weren't for patients, and I know you know this, but if it weren't for patients who show up at tertiary care centers like ours, meet physicians like me, and agree and sign consents that allow us to take their blood, to bank it upstairs, and to use that blood and those cells from that blood, which are invaluable because those are tumor cells, to understand the biology, that's what moves the field forward. It's the science that we derive from our patients' cells and their generosity in allowing us that sample and allowing us access to their data that helps us understand how to make the next generation of therapies that will target that stem cell.
Right. I'm a big believer in that. I go to a tertiary center, UC San Diego, and there are always those extra light green top tubes of blood that are drawn. And I say, what's that for? And they said, oh, that's for research. And they say, go do it. I'm willing to give the blood, and I'd recommend that to people. So, okay, so transplant is a serious discussion for a select group of patients, but the field is moving to say someday, can we have what you call the medical therapies, basically, where you can take a pill, or can we shut down the disease in some other way? Okay, one other area I want to go to before we sort of go through the laundry list of some of these drugs to just understand what they are is progression. So we have some people watching who may not have myelofibrosis. They may have polycythemia vera, or they may have essential thrombocythemia. But as they learn, they know that there can be progression from one to another. They're not on a JAK inhibitor. They might be someday. Um, and so, what about that? What do we know about um, slowing progression or even knowing who will progress?
We know that the disease is chronic and progressive. And as you pointed out, progression is not just a fear of the patient, it's a reality that we as physicians try to risk stratify. We try to understand when we meet patients, are there risk variables that may help us predict what that timeline might be in order to maybe make decisions, whether it is transplant or enrollment in a clinical trial or use of specific agents to try to change that rate of progression. And the rate of progression and the reason patients progress is we believe due to what's called clonal evolution. It's really a clone of blood cells that acquire more mutations and more alterations that allow that cell population to behave differently and to change the phenotype, the clinical picture. So we use variables like age, anemia, white blood cell count elevation, the presence of circulating blasts, low platelets, mutations, like I mentioned, high molecular mutations, or chromosomal abnormalities into prognostic scoring systems. And all of these things can actually be found online. Many patients will go online, they can find one of these prognostic scoring systems, they can plug their own information in and get a sense of where they fall in prognosis. But I will caution patients if you ever do that or speak to a physician, whoever speaks to it, is you have to understand statistics. When you look at that, please don't make the mistake of assuming that median survival that you see is going to tell you what your lifespan is. It doesn't work that way. But what it does give us is just a framework to understand where in the spectrum of patients you fall, and that might help us make treatment decisions about what might be the most appropriate therapy. I do think our approach is going to evolve over time to a more refined risk adapted approach where we use mutations to guide us not just in the selection of therapies, but the timing of when to use those therapies. But I can appreciate from seeing enough patients that progression is a real concern. And it could be a concern for an ET patient, a PV patient, and obviously for a myelofibrosis patient. And as you and I have discussed, the ultimate concern about progression is the potential to evolve into an acute myeloid leukemia, what we call an AML. Sometimes you might hear it called blast phase. And that's an aggressive form of leukemia that is problematic and obviously is a fear factor for patients and physicians treating patients with MPNs. And things that we look at to see if that's a concern are mutations like P53, a type of mutation, or already the presence of circulating blasts or blasts, which are leukemia immature cells in the bone marrow. That kind of information can help get a sense of what the risk may be of a patient transforming into an acute myeloid leukemia.
Let me just ask about AML for a minute. I know there's a percentage of MF patients who traditionally would progress to AML, and you look at these blasts. There's been progress in, I guess, what you'd call primary AML, people who haven't had myelofibrosis or anything, and there have been a number of drugs developed. But secondary AML that would come out of myelofibrosis, I understand has been more difficult. Where are we with that now?
So you're absolutely right. What we call de novo AML or primary AML, we have I would use the word plethora of different agents. It's not still not enough, but we have agents that can be quite effective in trying to control that type of AML and induce a response. Those agents typically are not as effective and don't have a very significant impact in the disease process in secondary AML that arises out of an antecedent myeloproliferative neoplasm, whether that's ET, PV, or myelofibrosis. So MPN-related AML is molecularly distinct from de novo AML. It looks different from a mutation profile and it behaves differently. And unfortunately, it is often more resistant or refractory to the traditional types of therapies that we give in that setting. And a great example is we do no benefit to an AML patient who had an MPN previously by giving them induction chemotherapy. Unless that's followed by a transplant, every study you look at tells us that that does not really improve the outcomes of those patients. So we know that's not effective now. We rely on drugs like hypomethylating agents, drugs like decitabine or Inqovi or azacitidine, Vidaza. These are epigenetic therapies. These are therapies that try to induce maturation of these immature cells. That's what leukemia is, or immature cells that don't know to grow up. We try to induce that maturation process so that they die a more natural death. It's less intense, but it's actually more effective with this type of AML. And of course, there are a lot of other therapies that are under clinical investigation. Some of them are molecularly directed therapies, like we've run trials with IDH inhibitors. These are oral drugs that specifically go after mutations that can be present in AML that arose out of an MPN that can be quite effective. So they're understanding the nature of that AML, the molecular profile could inform treatment decisions. And the reality is that's an AML that's problematic, and that our real goal is to stop the process from evolving to AML, because we know that treating that AML is complicated.
Okay. One other current therapy I wanted to mention, because I know even in New York it can be used a good bit, and there are many patients who are proponents of it, is interferon. We've had interferon for a long time. You haven't mentioned it, but where does that fall in related to either the treatment of myelofibrosis or PV in particular, maybe some of the other conditions?
So interferon, which is a biologic, which has been around a long time, is a really interesting compound. And there is a lot of data both in the lab and in patients who've been treated prospectively on trials, particularly polycythemia vera and essential thrombocythemia, that this is a drug that can be anticlonal. I use that meaning getting at that stem cell pool that's causing the problem, is quieting those cells down so that you can see changes not just in blood count numbers with ET and PV, but reductions in mutation levels that are driver mutations like the JAK2 mutation. And there are groups that have shown that reduction in those levels correlates with a better event-free survival, EFS. Events are clotting, bleeding, progression, and death. In ET and PV, so subtypes of MPNs, we really see the value in that setting. And I think that that's increasing in its utility and its use throughout the world. Myelofibrosis is a little bit different. There's probably some value there, particularly in treatment naive early forms of the disease, or what we call pre-fibrotic forms. And that's actually under active clinical investigation in a global study, taking patients with lower risk disease that maybe don't have so much complexity and so much fibrosis in their bone marrow, and have seen so many different types of therapies. Because I think interferon works best early on in a disease course. Once the disease gets too complicated and too advanced, interferon may not be that effective.
Okay. We mentioned at the outset about the ASH conference, and you have the European Hematologic Society, the EHA meetings as well, and other meetings that you go to. You speak at a lot of them. And here are some of the names that come up, and many of us have never heard of it. You mentioned selinexor. We've heard of that, and I'm aware of its use in myelofibrosis, rather a study in myelofibrosis, but for multiple myeloma, another blood-related cancer. You mentioned navtemadlin, if I got it right, an MDM2 inhibitor. There's another one, nuvasertib, I think, and another one called elritercept, and then there was anti-hemojuvelin antibody, and one of them under that category is DISC071. They have numbers when they don't have names. So help us understand this sort of constellation of what's going on and what you're trying to figure out there, and where even being in a clinical trial for one of these drugs could be beneficial for someone.
So, what you'll notice certain themes, and I think it is particularly for the uninitiated, it is somewhat frustrating to see or hear these names because it really doesn't resonate. I think if one were to put that aside and just think about themes, it makes sense. So, for example, a lot of the themes of what we're trying to do in translating the understanding of the biology from the laboratory into the clinic is targeting either signaling pathways. So these are pathways that are inappropriately activated in malignant cells that continue to tell them to proliferate, to secrete inflammation, to do things that they're not supposed to do. So our cells all have these very intricate and complex and overlapping signaling pathways. In many malignancies, including myelofibrosis, multiple signaling pathways are inappropriately activated. They're hyperactivated. So for example, Jakafi and the other JAK inhibitors inhibit the JAK-STAT signaling pathway. And you'll notice those drugs end with NIB, inhib. So like those are small molecule inhibitors that inhibit enzymes in those signaling pathways. So nuvisertib is a small molecule inhibitor, which is a selective PIM1 kinase inhibitor. It inhibits an enzyme in a signaling pathway that is very relevant to the biology of the disease, and has nice data so far that shows reduction in symptoms, particularly, but even spleen and cytokines as a single agent in patients who've already been on a JAK inhibitor. Other drugs may be going for trying to turn on mechanisms that have been turned off. And one of the, again, I'm going to go back to navtemadlin, but it's the same with selinexor. And what these drugs are doing they are focused on different pathways, but namely one unifying theme, which is the p53 pathway. It's the master regulator of cell fate. And in a normal situation, your normal cells would get cues if they were infected or corrupted in some way to die, to commit suicide, and that is governed by the p53 pathway. So it's an intrinsic pathway that limits the cell's life. The problem is malignant cells have co-opted that system, and they've turned it off, and they turn it off in different ways. Both navtemadlin and selinexor, through different mechanisms, actually act on trying to turn that pathway on, trying to tell that cell, encourage that cell to die, actually, that something is wrong. You need to use your intrinsic pathways and to die. So those are examples of turning pathways that have been quiet, that are beneficial, turn them back on and kill the cell. Then you might see a drug like the calreticulin antibody, and that inevitably will get a name. And you'll notice early on, it's just letters and numbers. And then as the development goes on, then it becomes funny names that don't make any sense. And then ultimately, when it gets to the commercial space, it will be, a catchy name that can be marketed. But the drug by Incyte, that's a mutant CALR antibody, is a perfect example of what will be a MAB, a monoclonal antibody, that is targeted at the CALR mutant protein that's expressed aberrantly on the surface of the cell and is a marker for those abnormal cells. And what better way to attack an abnormal cell than to have a selective marker? It's like a handshake. The cell is showing that abnormality. They're saying, here I am. So these guys invented a drug. And it's not even a drug, really. It's a peptide. It's a protein that just goes and binds that. And by doing that, internalizes that protein complex. And that leads to the death of that cell selectively. So it's approaches like that I think will be very fascinating. So those are immune-based approaches, and they have bispecific T cell engagers that multiple companies now are developing that introduce T cells in a myelofibrosis patient to the abnormal cell by having two ends of that antibody, one that goes after the CALR, for example, and the other one that goes after CD3 on a T cell, and introduces the two cells together so that that T cell does what it should have done in the first place, which is recognize and create a response to that abnormal cell. So what you'll see is drugs that get into the cell that affect pathways, drugs that get into the cell that try to turn on pathways, and then drugs that try to use the immune system in different ways to go after the abnormal cell. So, there are these general themes that one can start to appreciate. Funny names, but rational themes.
Okay, so that brings us to a discussion about clinical trials. So these are drugs that are being studied at different levels. You're involved in a number of studies. Other major medical centers are, both in the US and in Europe, other places. For people listening, what would you say to them about considering being in a trial? Once there's a clear picture of their myelofibrosis, their PV, whatever it may be, there's a clear picture of what their case is, if you will. And is there one of these inhibitors or stimulators or antibodies that could line up with that. Take us through the thinking and the discussion between a doctor and a patient about considering being in a trial.
So, I think I have to first acknowledge that being on a trial is, I think, scary. It's scary because you feel, and I hear this all the time, guinea pig. You feel as though you may be experimented on. And I always try to caution patients from that mentality because, I think if you have a disease like myelofibrosis, for example, that is, obviously frightening to have in itself and can limit both the quality that you have and maybe even the time that you have, then it warrants consideration for trying to do better than what the standard of care is. Because we know the standard of care, which is mainly JAK inhibitors, has benefit for sure, but it's incomplete for many patients. So I think the consideration for trials really hinges on the patient's goals and desires. And clinical trials may not be reasonable for every patient. First of all, geography influences clinical trials. So if you live in a rural area where maybe the distance between you and a center that has a clinical trial may preclude you from being in a clinical trial. And we know other things get in the way of clinical trials: language barriers, cultural barriers, financial barriers. There are a lot of barriers that could get in the way of a clinical trial. But I would encourage any patient who has the ability to see, and obviously this is self-serving because I'm going to say who sees a specialist and goes to a tertiary care center because that's typically where the trials are, should seek at least a consultation to understand what does someone who does this full time think about their disease and clarify or classify their disease, and what clinical trial options exist. I've been around long enough that when before Jakafi was ruxolitinib, it was INCB 18424, and that was a drug that we used in the clinic, and we just prayed that it was going to really make a big difference, and it did. Not enough of a difference, but it has made a big difference. And I remember those brave patients that were the first patients to get on that study. And I think that they did it because they set the tone for the whole field and the development of other therapies, and because of what they put themselves into. And those patients needed the therapy, but what they did allowed us to prescribe the drug for a lot of patients. So it has a profound impact, way beyond that individual. But the reality is, patients go on trial for themselves, and that is what it should be. But you do end up helping the greater good too at the same time. And I think you have to feel comfortable with it. You have to read the consent form. You have to ask the questions: what does it involve? What are the potential toxicities? What would be the consequences of participating in a trial? I feel very comfortable saying in 2025, any trial I'm aware of that's offered to patients is an informed trial. These are not trials where we're taking random agents off a shelf and saying, well, I'm going to throw it over here and let's hope that this works. I mean, these are drugs and agents that are vetted, that have rationale, that go through many layers of pre-testing before they go into humans. And you have to trust your physician, because if your physician is working with you, then the point would be that the trial that's offered to you would hopefully make sense. And trials should make sense. You have to ask those questions so that you understand what you're getting involved in.
Okay, a couple of questions about clinical trials. One is people, you mentioned about the people say, am I going to be a guinea pig? They also say, am I going to get the good stuff? In other words, are you going to give me a sugar pill? And it's somebody else in the waiting room who's getting the good stuff. I'm not. Maybe you could talk about that related to these kind of trials for these drugs that are investigational now.
Yeah, so if you're in a phase 3 study that's randomized, then it is entirely possible that an arm of that trial, so a computer randomizes you to arm A or arm B. Arm A may be the active, clinical trial agent. Arm B could be a placebo, but that has to be disclosed in a consent form. So that's essential. Is you can ask the physician, well, do I get the study drug, or is it going to be randomized? Is it going to be placebo? But placebo is not always bad. So for example, in the Manifest study, or right now that's currently enrolling in the Sentry study with selinexor, you get randomized as a JAK inhibitor naive patient with myelofibrosis to either Jakafi plus a placebo, so you're still getting active therapy, you're not getting nothing, versus Jakafi plus selinexor. So the idea is, can we build upon the success of the standard of care? So that becomes important. It would be unethical to give someone who needs treatment nothing. It's not unethical, and it's practical to give someone who needs treatment what we would normally give them, what would be standard of care, plus a sugar pill, which is blinded, versus standard of care plus the study drug. Ask the question, which one is better? And you'll notice that most of the trials, and you can ask your physician, most of the trials will allow crossover, and that becomes important, which means that if you do get that placebo, which the physician won't know, you won't know, but a computer will know, at some interval, most trials will allow you to, if you are on the placebo, to cross over to the active compound. And that becomes important. So it becomes more of a question of, do I get the active combination up front or is it delayed? So that's a nuance that I do want to stress. If you're participating in a phase 1 or phase 2 study, you're going to get the drug. Those are not placebo-controlled studies. So you should always be getting the drug in those settings.
One other last question about trials. Some of us have other conditions. Now, it could be diabetes, could be a heart condition, high blood pressure, whatever. For me, it's actually a fortunately pretty well controlled leukemia, chronic lymphocytic leukemia. But we're often excluded from trials, depending upon the condition, because we understand the worry of the investigators. Is our data kind of clean? Can you extrapolate from that? So talk about that for a minute, about exclusion criteria, and also this term I've heard sometime about compassionate use.
So the reality is there are biases in the way we do trials. So we do avoid patients that may be inappropriate for a trial. And these are real patients that might be in need, but the trial may not be appropriate. So these can be patients who have very significant kidney dysfunction or heart dysfunction, or might have a competing malignancy. And interesting, CLL, which is more frequently seen with patients with MPNs, has different stages. And sometimes studies do allow patients who have very indolent type of CLL, do allow them to go on study. But often trials will be very particular, particularly trials that are registration studies where they're going for an approval. Because the last thing a study needs is to have confounding data where they're unclear whether they're making something that's unrelated to the myelofibrosis worse, or that condition is making the assessment of the drug harder to appreciate. So there is a tendency to exclude patients who have extremes of comorbidities. Not all comorbidities, because to some extent, heart failure, if you have grade 1 heart failure, that's often allowed on study because that's a reality of life. So there are very strict inclusion and exclusion criteria, what we call eligibility criteria, that determine the ability to put someone on study. And most of those are there for safety reasons, to avoid causing additional problems with a drug because it's not intended for that reason. But I will say that the other reason why they're there too is because if you have a very messy patient population with lots of diversity and heterogeneity in organ function and et cetera, it can be very difficult to assess whether the treatment we're giving is safe or effective. So to some extent, we do create out of necessity some homogeneity out of the heterogeneity.
All right, and just the word about compassionate use. If, let's say, somebody would otherwise be excluded, but could their doctor make a plea for maybe a late stage patient, hey, this really might save their life?
Yeah, so compassionate use, I used to make the statement, I make it much less today because it falls flat, but compassionate use is often not that compassionate, meaning it is trying to get access to a drug for an individual under what's called an IND, an investigational new drug, for just that one. So it's creating basically a protocol for one individual. And what is not always appreciated is that sounds great and it sounds obvious, but there's actually many steps that go into that require a lot of regulatory oversight and approval and effort, which is not provided by a company or anyone. It's just the physician and whatever team members he can assemble to try to do that. So although compassionate use is an opportunity to get a drug, it's often an opportunity that's plagued by the realities of a very cumbersome bureaucratic system that doesn't actually make it very easy to get to that drug and timely to get to that drug. So although it is there, and in some cases it is helpful, it's not always practical or realistic that people can get compassionate use drugs. And sometimes the access to those drugs are restricted by the FDA and or the sponsor. But compassionate use can sometimes be an opportunity to get a drug that wouldn't otherwise be eligible. And that's a key thing. If there's a trial that allows access to that patient, they often will not provide compassionate use because it would be felt to be unethical to allow someone to get access to a drug while all other patients would have to go through the normal routine of the clinical trial. So compassionate use really has to fit very strict criteria, and is not always achievable.
Okay, thank you for explaining that. So, okay, we have ASH in the rear view mirror. You have other conferences during the year. You're speaking at a number of them. You're involved actively in research and seeing patients as well. So, for our audience, John, I want to talk about two more things. First of all, I want to get where your head's at as far as, I hate to use the word excitement, but promise in the field, how you feel about it, and then what people can do about it, okay? Because our listeners are saying, okay, well, Andrew and Dr. Mascarenhas covered a whole waterfront here. What do I do with this information? So first, how do you feel about it? What's the headline for you?
The headline in terms of where we're going in 2025?
Yeah, where we are and where we're going.
Well, I mean, I think we are way further ahead than we were in 2005 and in 2010 and 2015. And I've watched that grow in terms of our understanding the amount of attention and support that's provided to this rather small area of hematology, both from the federal government in terms of NIH grants, in which we indulge in getting to try to help move the field forward, independent of pharmaceutical interests, but as well as pharmaceutical interests, and as well as philanthropy, that all helps grease this machinery of translational research and understanding. So we have cadres of these really smart, well-intentioned PhDs and MDs and MD PhDs that are helping us understand the biology, and that is directly translating into the clinic to help us fine tune our treatment. So you look at ASH or EHA today, and you see endless abstracts of agents that we didn't even know about, or targets that we didn't even understand even 10 years ago. I have to believe that that's going to translate ultimately to better therapies for our patients, and multiple therapies, because I don't think it's going to be one therapy fits all. So it's really trying to understand how to get access to and develop multiple therapies that could be used in different subsets of patients or in different combinations. And that is something that I do feel optimistic. And I would say, if the banner is it's moving forward in 2025 and beyond in rational combinations and allowing science to drive the advancements.
So you're feeling positive.
Yeah, but you've known me a long time. I always feel positive. I am definitely feeling positive. I walk from 80th Street, where I live, to 100th Street, where I work, and I'm enthusiastic every day I come in, because I do believe in the mission, and I do think that we're making strides. I see it. I see it in the patients that I treat, but I believe it for the future.
Okay, so then we say, okay, current patient, what can I do now? Maybe somebody has a lot of anemia, or somebody has platelets they're worried about watching that are lowering, they're getting bruising or bleeding or whatever, and they're saying, this is what I'm dealing with right now, or my spleen is enlarged, or whatever it is. What are things that people can do now that could make a difference in the short term, even?
Well, I think if you're having spleen or symptom burden, and particularly if it's interfering with your quality of life and your activities of daily living, the obvious thing is you should be seeing a physician who specializes in hematology, um, ideally someone who has expertise in this area, and a JAK inhibitor is the frontline therapy to try to address those aspects. It doesn't preclude one from looking at clinical trials that might even be combinations upfront of a JAK inhibitor plus an active agent to see if one could get even better, deeper, earlier responses. But I think at the very least, a JAK inhibitor to try to address things, because you get no extra points or rewards for suffering through a big spleen or symptoms. And in fact, it's harder to rescue those responses if one delays. And the data is very clear about that. The longer one delays starting therapy, the less likely that therapy will be effective, and the less likely that the effects of that therapy will be durable.
Mm-hmm. Well, you alluded to it, and I'll say this just directly. I really believe, and you may live at a distance from one of the major centers where there is an MPN specialist, but there are MPN specialists. You refer to cadres of researchers and physicians now who are working in this area. And I think it really makes sense to have a consultation with somebody like Dr. Mascarenhas at Mount Sinai, my doctor, Dr. Jameson in California. There are many others, fortunately, who are studying this and working on it and understand these nuances so you get personalized care for where you are now and where you may be headed. John, did I get it right?
I mean, I would say spot on, spot on.
Okay. Well, this has been a really helpful discussion. Is there anything you wanted to add, John, that you think we didn't cover?
No, I mean, I think we covered all the major points. I think I conveyed my optimism, my continued optimism. And I maybe just the one last thing, which is I think one boon that we have in this field is it's a very collaborative field. So what you'll notice, and you can just look at abstracts and trials and studies, and you look at all the names that are listed with it, it reflects a collaborative, because these are rare diseases, you can't work in a silo. So it reflects the collaborative nature of this field. We're all friends and colleagues. We all have a unified mission, and we're really focused on that as an international team. And I think that's why I remain enthusiastic that we're going to make the progress.
Okay. Well, thank you for your collaboration and people, and that I'm seeing a lot of younger physicians and scientists interested in this area. Yay. So that gives us a great deal of hope. Dr. John Mascarenhas from the Tisch Cancer Institute in New York, thank you for your devotion to us. And just go for it. And we really appreciate your time. I'm Andrew Schorr in California. Dr. Mascarenhas is in New York. We're all bonded in this together. Remember, knowledge can be the best medicine of all.
I don't know about you, but that discussion was the definition of news you can use. Thank you, Dr. Mascarenhas, and our patient advocate and moderator, Andrew, for taking the time out of your busy schedule to keep the patient story community informed. If you are a patient, caregiver, partner, or advocate, consider being a voice leader in your community or with us here at The Patient Story. To wrap up this discussion, I want you to leave with the important takeaway that to be empowered is to be inspired. We want you to make those informed decisions about your care, and that includes being educated about the latest treatment options. A reminder, we want to hear from you. A survey will pop up when the program closes. Thank you again to our sponsors, Sobi and Insight, for their support of our independent patient program, and to all of our promotional partners. Until next time, I'm Tiffany Drummond signing off, and on behalf of The Patient Story, thank you for watching.